You finished just on time. That's fantastic. Unfortunately, we don't have time for questions. So Rick is going to set up the next presentation, which is by Professor Gary Schenk from the School of Chemistry and Molecular Sciences, and he will be talking to us about bioleaching for in situ leach applications.
Yep. Hello, everyone. I just quickly share, I guess, the screen. Hello, everyone. Very happy to be here and present sort of what we've been doing. Also quite honored that I can fill in for Alice, who was supposed to give this talk but couldn't be here today. So a fairly big pair of shoes to fill for me. So I try to give it my best. So the aim of our team is to develop, in a nutshell, bio-based methods that can be of interest to the mining industry. And the particular focus has been the development of enzyme sort of based methods, in particular in the leaching, copper leaching process, to be very specific. Now, before I give you sort of background and updates, should introduce you again to our team. Not that I'm going to talk about each and every one of these wonderful individuals. Most of them you all know quite well. Bottom line is it's a very diverse team across the university and also with colleagues from other universities. And amongst ourselves, we sort of cover a fairly broad range of disciplines. And that's also quite essential to sort of develop sort of bio-based technologies in an environment that is at first sight not all that sort of amenable to biology. Now, Alice, of course, is the coordinator of our sort of team, but we've been sort of working together quite well for a couple of years by now. So what is our motivation or our sort of starting point? So what we have been interested in is in particular the bioleaching process. And I mean, I had the pleasure, I know Mansour quite well, so we've been talking on and off for a number of years, sort of how to optimize our sort of collaborations. As a particular sort of area in this bioleaching field, what we try to do is we try to sort of come up with a way how we can improve the way, in this particular case, say copper can be leached from sort of environments or ore such as chalcopyrite. I mean, it's a quite well-known sort of process whereby iron 3 plus here is needed in essence to drive the oxidation of the copper in the ore, which then leads to leaching of copper in the 2+ oxidation state. In the process, iron 2 is formed, which then is recycled and starts the whole process again. So that's not really new at all. And in essence, we need iron 3, and for that, we don't actually need biology either to produce this. And in our lab as well, and we're not the only ones who've ever done that, but for instance, in Paul Bernhard's lab in our school, he set up this simple sort of galvanic cell here, whereby he, in this particular compartment, he had a platinum electrode here on the right-hand side. I might actually change the pointer. You might actually see that a little better. There's a pointer here. So you can easily sort of oxidize a ferrous ion. Say, for instance, if you have ferrous sulfate in this compartment here, you can oxidize this to iron 3 in this particular setup that sort of leads to the formation of hydrogen gas in the other electrode. And if you run this over a number of hours, I mean, as I said, that's not rocket science. You can sort of monitor sort of the concentration of iron 2, which gradually is sort of depleted. And at the same time, you start to make, of course, iron 3 as you oxidize the iron 2. Now, what Paul did as well, and Jamie, the research assistant in the group, basically you can use this iron 3 and, for instance, you let it react with chalcopyrite. And if you let that happen for a number of days, like they did that for roughly a fortnight, you gradually see sort of course, a buildup of copper that accumulates in the solution over sort of 2 weeks or so. What they could measure is that up to 70% of the copper that was sort of encapsulated in the crude raw material, the chalcopyrite, eventually leached into the solution. Now, what we were interested in, sort of in parallel of doing this, is, well, can we develop a more efficient, cheaper, more greener, environmentally more friendly way to generate the iron 3 that's obviously needed to drive this process? And that's sort of where the biology comes back in the bio sort of leaching process. So we looked again at arguably the best known bioleaching sort of process from an organism called Acidothiobasilus ferrooxidans. And I apologize, I'm not a microbiologist, so I'm struggling with these words still now. So this Acidothiobasilus has sort of this particular sort of mechanism localized in its membrane. It's a mechanism that's based on a number of proteins that catalyze the oxidation of iron 2 to iron 3. And in the process, the electron that's being released from this process is sort of shuttled or carried, if you like, through a sort of electron transfer chain, whereby ultimately oxygen is reduced to water and some sort of so-called reducing equivalents, NADH or NADPH, are being generated. So this pathway is an essential pathway for this organism to gain energy, in essence, to survive, to live, to thrive. So that's basically its survival is linked to the oxidation of iron. So obviously that's a very effective sort of process for this organism to thrive. Now, the problem is in bioleaching quite often, and in microbiology, in more general terms, of course, if you play with microorganisms, they have to live, so they have to be sustained, maintained, and they can be quite vulnerable to changes in the environment, changes in the amount of nutrients that are available, the temperature, the pH, other sort of environmental factors. So we were thinking it might be very useful in terms of control and potentially also in terms of effectiveness if we could run this process of enzyme-driven oxidation of iron by just relying on the enzymes and forget about the cells around them. So basically, worry just about the enzymes, the proteins that do the job, and forget the rest around it. Now, before I might sort of talk more about enzymes, I'm aware that not everybody in the audience has sort of worked with enzymes or knows too much about them. So I might just give you a very, very quick sort of rundown what they are or what they're used for. Now, in simple terms, an enzyme is a biological catalyst. That's all it is. Basically, enzymes are catalysts that are essential to drive pretty much all the chemical reaction that make biology possible. So without enzymes, there will be no life, so to speak. But by and large, it's really just a catalyst like any other catalyst as well. Now, how does it work? Excuse me. It's not Corona, I can promise. So when you think about a chemical reaction, any reaction, that can be the oxidation of iron 2 to iron 3 or any other kind of reaction, they all sort of basically proceed via a so-called activation barrier. So in order to proceed, a chemical reaction needs a push, has to basically overcome a certain energy barrier in order to proceed to products. Product in our case could be, for instance, iron 3, but the same is for any other reaction of relevance. Now, what the catalyst does, enzymes included, all they do is they lower the height of this activation barrier. And by doing so, of course, you need less of a push. That means the reaction can go much faster. So that's what a catalyst does. That's what an enzyme does. It makes reactions go faster. Now, in biology, enzymes have adapted to all sorts of environments. I mean, like certain bacteria live in very extreme environments, extreme in terms of temperature, extreme in terms of pH, metals, for instance, and so forth. And you find that microorganisms have adapted to actually utilize whatever the environment throws at them, uses enzymes to basically thrive in such environments. Now, the good thing also is we have, in the meantime, developed a whole range of sort of engineering techniques that allow us to work with enzymes as if they are just any other sort of chemical sort of compound. We can make them look different. We can make them work faster. We can make them be more thermostable. We can make them work on different reactants and so forth. So they're very malleable. We can engineer them. We can also optimize temperature range and pH range where we can employ them. So they're very real, sort of good agents in biotechnology, full stop, because they can be used to carry out reactions of interest. Of course, in our case, the reaction of interest is to catalyze the oxidation of iron 2 to iron 3. Now, the question, of course, is which enzyme? Obviously, there are many enzymes, which one should we pick from? And in essence, there are most likely many, many different candidates that are suitable for a particular application, like, for instance, the oxidation of iron 2, or any other sort of reaction you may think of, like, for instance, in Mansour's field of work, if you're interested, for instance, in tailings. For instance, you may want to sort of find enzymes that have been optimized, for instance, to, for instance, engage with or bind gold or platinum or any other sort of metal in there. That all exists. You can find them all. Enzymes are extremely sort of versatile. So in our case, we thought as a good starting point is, well, rather than sort of basically go shotgun, let's look at a process that's well understood. So we went back to the acetylthiobacillus ferrooxidase process and have a look what kind of enzymes are involved in this particular process here. Of course, you may rightly think, well, the most likely candidate or interesting candidate to work with is the very first enzyme in this chain, which, I mean, the label here, the cryptic, what it stands for is cytochrome C2. That's the name for this particular enzyme here. That will be the obvious candidate you may want to work with if you just want to rely on the enzyme itself. Problem is, as you might see from this illustration, the enzyme is actually embedded into the cell membrane. So this gray thing here, that is the cell membrane that is very lipophilic. In other words, it's made up of fatty acids. So it's not hydrophilic. So in other words, this protein is not easily soluble, say, in a watery environment. Means it's not very easy to work with, not very easy to handle. We have been working with it, it's ongoing research, but we had to do a fair bit of engineering to make this enzyme more soluble. In the process, we changed its catalytic sort of properties. So we're still in the process of sort of further engineering the enzyme so that it actually works optimally in a soluble form. But while this was going on, we thought we're not going to rest on our laurels. And we thought, well, how about the next enzyme in the chain? Now, the enzyme is called rustacyanine here. Now, this had a few obvious sort of advantages for us. The first one was it is soluble. So it's a water-soluble enzyme, which means we can easily manufacture it, produce it, and work in water, which we want. We want to basically work in a watery environment. The enzyme also was studied previously, so it was not new to us. There was actually quite a wealth of literature available. For instance, the crystal structure shown over here was known. It's actually a copper-dependent enzyme. It needs copper in order to work. So it has one copper bound in what we call the active site here. And so it was a very good target for us to work with. So what we did was we managed to sort of produce the enzyme in large quantities using E. coli cells. So we could isolate it. It was stable enough. We could produce large quantities. And the first thing, of course, we wanted to do is to establish, can this enzyme actually catalyze the oxidation of iron 2? And long story short, it can. So this is sort of a typical kinetic assay where we measured the catalytic rate here on the x-axis as a function of having more and more of iron 2 present. We could easily measure, of course, the reaction rate, because what happens in the reaction is, remember, it has a copper in there. As we actually oxidize iron, the copper in the enzyme gets reduced to copper 1, and then it has to be regenerated to copper 2 to keep carrying out the same reaction over and over again. Now, what we could establish is the enzyme is, I wouldn't say it's a rocket, but it's not the slowest catalyst either. So basically, one enzyme can turn over 60 iron 2 ions to iron 3 per second. So one enzyme turns over 60 iron 2s into iron 3s. Reasonable rate. Not fantastic, but better than nothing, I dare say. Bottom line is, it worked. Excuse me. So next thing we did is we want to check if indeed the enzyme can be used reversibly. Remember, a catalyst, they're very effective because not only they're fast, but they can also be regenerated. So one enzyme basically does the same reaction over and over and over again, but it has to be, of course, regenerated after every single catalytic cycle. So doing cyclic voltammetry, our electrochemist in the group would demonstrate that the enzyme can reversibly undergo numerous cycles of oxidation and then reduction again. So in other words, as the enzyme gets reduced, when it carries out its catalytic function here, it can then be regenerated oxidized, so to speak, ready to make it ready for the next catalytic cycle. So that was all good. So far we were on a winner. We had an enzyme that could do the reaction and which also was rechargeable, so we could use it time and time again. And that was also in reasonably good agreement with what we knew from the literature. The problem sort of emerged as we started to test the stability of the enzyme. Now, there was very little literature available and in parts also contradictory literature. So what the group did, Paul and Jamie in particular, they started to sort of basically look at the stability of the enzyme as a function of temperature. So basically, starting off at room temperature, 25 degrees here, and we slowly sort of went down all the way down as to 5 degrees Celsius. And we had a look what happened. Now, what we observed was that gradually, if you keep sort of recycling between the oxidized and reduced state, the enzyme gradually sort of does deteriorate, does degrade, basically falls apart. Now, that happens quite rapidly, rapidly at a reasonable rate at room temperature. You can slow it down significantly as you get to colder and colder temperatures. It's it's most stable at 5 degrees Celsius. Now, that was a little bit of a damper because think about it. I mean, we like to actually use sort of, like to generate such enzyme sort of driven iron 3 for the leaching process while under ambient room temperature, or at least not very cold temperature, because that would be from the energy point of view, not very practical, because we would have to somehow set up our system so it's always refrigerated. In Australia, for instance, that
2 minutes, Gary.
Okay, I'm almost done anyway. So basically, the bottom line was the enzyme works best at low temperature, which might be useful in Canada, not the best in Australia. So it was a bit of a dampener. So we have an enzyme that works, it's reasonable, but it's not optimal in terms of temperature. Now that basically meant we had to go back to the drawing board. We have a number of options. One is, of course, we can engineer the enzyme to become more thermostable, but a more practical approach, which we do in parallel, is we are searching or discovering novel leads, novel enzymes other than rustacyanine that can actually carry out the function as well. So that's where we are at. So to put it all together, it's the last slide. So that is just to summarize where we are at and what we are aimed to do. So basically the central party that I'm circling, this is sort of where our enzymes come in handy. Eventually, when we have the best catalyst, is in essence producing the iron 3 that is then used in the leaching process, and then it can be all recycled here again. So good news is we could demonstrate enzymes can do this. Damper is that we haven't found the best enzyme, or at least not the best engineered enzyme. So that's ongoing research. But yes, I think that's where it is. So if you have any questions or comments, of course, you can always ask me. But Alice is sort of our focal point. Thank you very much.
Thank you, Gary. We've got 1 minute left. Rick, are there any questions in the chat? No?
No, it's not a question in the chat. It's a chat. It's a response and answer.
Okay. Does any... Go ahead.
If I can get this. Can you hear me?
Yes.
Yeah. Rick here. So so what's the next step in the program?
A few. So one is obviously engineering or work further with the rustacyanine to work to make it more thermostable. Another one is we are sort of in process using bioinformatics to identify other candidate proteins that we can sort of integrate in the program. So that's basically mainly bioinformatics, where we sort of look at metagenomic and also, when available or if available, metatranscriptomic data to find candidate enzymes. And thirdly, didn't speak much about this. I did mention we work on this cytochrome, which by itself, we had to modify it so it could be soluble. We are sort of slowly getting to a form that is catalytically quite attractive. In other words, we had to engineer it so that it works in solution the same way it works also in a membrane-bound form. So these are the three major areas sort of we are driving at the moment.
Thank you, Gary. Thank you very much. I think we need to move on, and I would love to introduce Dr. Eleanor Lebra, who will talk to us about the complex all bodies database.